So welcome everybody to this week's seminar. Happy to have you here. We have a number of people from all over the place. We have next week's speaker from Chile who's joined us, which is pretty cool. So we have Spain and Chile connected together. We have someone from Pittsburgh. We have a bunch of local old retired people around here, and a bunch of faculty members from the University of Oregon, as well as the students. So a good audience. Thanks for coming. I'm really delighted to introduce our speaker today. Our speaker is Maria Elsa Vasquez Otero. She has 4 names.
Too long.
And Elsa comes to us from the University of Vigo, where she is a professor. I know a little bit about her, so I'll just share what I know. Elsa actually was born in the northwest corner of Spain in the Coruña area. And I asked her one time how she got started in marine biology, and she tells me that her father used to take her to the ocean all the time. And she practically grew up in the ocean, on the rocky shores of Coruña, I suppose, snorkeling and swimming and sitting on the beach. And she can correct me if I'm wrong about any of this when I give her the.
No, you're right.
But eventually she ended up doing her college work also in the same region, and went to a university in a very famous city, the University of Santiago de Compostela. And I don't know if you've heard of that, but if you were a practicing Catholic, you probably would have heard of it, because it's a very, very famous town with a big cathedral where the brother of Jesus, St. James, is supposed to be buried. And there's been a site of pilgrimage since the Middle Ages. So there's a famous trail to Compostela that ends at Elsa's University, more or less. She did her undergraduate work there. She also did her PhD at the same university, and then received a fellowship to come to the United States. And amazingly enough, she came to work in my lab. She had become very interested in a group of animals called ascidians, sea squirts, tunicates. And I happened to be working quite a lot on ascidians back in those days. And Elsa actually was very, very well trained as an ascidian taxonomist. She had become sort of the heir apparent for the world of ascidian taxonomy by working in Paris. She had gone and spent some time at the National Museum of Natural History in Paris, the famous museum where Cuvier was, and Buffon and all of those people way back when. And they had the best ascidian taxonomists in the world there, husband-wife team, Claude and Francoise Monod. And she worked in their lab, learned how to do the dissections and the preservations and everything properly, and then wrote to me and asked if she could come and work in my lab. And I readily said yes. And then she might not know this, but I forgot about it. And I forgot that she was coming. I hadn't really paid attention to the arrangements. And one day she showed up at Harbor Branch Oceanographic Institution in Florida, this exuberant little girl with long blonde hair sticking out at the sides, and said, I'm here for my postdoc. And I basically said something like, who are you? And anyway, it wasn't quite that bad, but more or less. I kind of forgot about it until the last minute.
We didn't have internet at that moment.
Yes. Anyway, Elsa spent I think about 3 or 4 years in my lab, right?
Yeah.
Maybe 4 years.
No, two years, and then I came back, and I keep on going back to Harbor Branch for summertime and 6 months, and
yeah. Anyway, she became a fixture in the laboratory, and it was a time when we had 4 postdocs, and they became great friends and all very productive people. It was really a great period of time. So during that time, I think she worked with us in the Bahamas. She was on the boat in the Gulf of Mexico when we first fertilized the eggs of tube worms from methane seeps and figured out that they weren't pogonopherans, but they were actually protostome annelids. Elsa was a participant in the Nature paper that described that. I think you went with us to Hawaii, did you, on a cruise?
Yeah.
We did, I guess, at least one cruise in Hawaii during that period of time. And then a bit later, we were invited by a colleague who had been a sabbatical visitor in our laboratory, Suki Ekaratna, invited me and Elsa and one of our other postdocs, also from Spain, to Sri Lanka. So we spent some time, several weeks in Sri Lanka, teaching the Sri Lankans about taxonomy and diving coral reefs and looking at archaeological sites. It was a good time. Anyway, that's enough for going down memory lane. Elsa is a professor at the University of Vigo. She went there right after her postdoc, and she's been there ever since for about 25 years or so. She worked her way up to full professor status, and I think also spent some time as the vice president for research at the university, which is a very important position for scientists, and lots of control. She's been ever since then, she's been traveling everywhere and working everywhere. She's done work in the Antarctic. She's done work in Chile. She's done work in Panama, in Canada, in Cambodia, some others that I'm missing, I'm sure. But essentially, she's a world traveler, very famous marine biologist, and I'm delighted to introduce to you today my good friend and colleague, Elsa Vasquez.
Okay, thank you. Thank you very much for this very nice introduction. It took me back very nice memories. And Craig forgot to tell you that when I told him that I was going to be married, he just sent me an email saying, if you invite me to your wedding, Robin and I will be in Spain for the wedding. And they came to my wedding. And it was a very nice day. Well, because not only because of the wedding, but because I had very nice good friends with me that day. So first of all, thank you, Craig and the Oregon Institute of Marine Biology for inviting me to give this talk. And of course, thank all of you for attending to the talk. I'm going to share the screen now. What do you think?
Yes, that's fine. I'd ask the rest of you to please turn your video off to help us preserve our bandwidth. Thank you.
Okay. Can you see my screen?
Yes, that's perfect.
Okay, so during this talk, I am going to present some results of a project to study the different responses of commercial bivalves to heat waves and low salinity events and their implications for small-scale fisheries. Of course, this is not only my work. Other researchers from the University of Vigo, Santiago de Compostela, Coruña, and South Carolina, there in the US, took part on it. And also we count on the help of the technical assistance of the fisher guilds, all of them marine biologists too. So I'm going to start presenting you the problem that we have, not only here in Galicia, but in other parts of the world. I will follow just explaining you our hypotheses, our objectives. I will go very short through the study site, and then I will spend most of the time just explaining some of the results that we have at the mortality and growth in the fishing beds of these bivalves, physiological responses to heat waves of the adults, and also the physiological responses to low salinity of adults and juveniles. And now I will finish asking you something. And now what? And since this question is going to be answered by you, please pay attention, and of course feel free to ask any time if you don't understand something. So let's start with the background. The regional climate projections from the European for the European coast, North Atlantic European coast, predict higher frequency, duration, and intensity of heavy rains events, and also heat waves. These cause not only massive mortalities of coastal species, but also sublethal effects. Let me see if I can, okay, now. Sublethal effects as physiological changes, growth decrease, or even reduction of the reproductive success that triggers changes in size, structure, and distribution of populations. And all these can have dramatical changes, biogeographical changes. And this is of special concern when the affected species support important fisheries with huge socioeconomic cost, provoking major impact and social alarm that normally is well reflected in local newspapers. Why this alarm? Because small-scale fisheries account for more than half of the world global fish catches, 2/3 if we just take into account direct consumption. They employ more than 90% of the fishers, and these fisheries are firmly rooted in the local community, because normally provide direct fish consumption within the community. And these fisheries take place in the intertidal or the near shore. Particularly here in Galicia, Galicia has supported important shell fisheries throughout its history. We have more than 1,200 kilometers of coastline, 70 million square meters of intertidal sandflats. We have 3,800 small fishing boats, not recreational fishing boats, but professional fishing boats that employ more or less 10,000 people with incomes of 100 million euros per year. The fishers are grouped in what we call fisher guilds, cofradías de pescadores, that are structures that come from the Middle Age, and they keep on doing the same. And these fisher guilds here in Galicia are located in very small villages where the artisanal fisheries are an economic important activity. In Galicia, there are 4 main commercial species. 2 native clams, Ruditapes decusatus and Venerupis corrugata. 1 introduced clam, the Manila clam, Ruditapes philippinarum, and a native cockle, Cerastoderma edule. These together, they represent more than 80% of the total bivalves landings, with a commercial value of 74 million euros per year. The 4 species live in the intertidal and shallow subtidal sediments, but the optimal conditions, environmental conditions, differ among the 4 species. The most tolerant is the introduced species Manila clam, whereas the very well appreciated Venerupis corrugata or the Ruditapes decusata have less tolerance to salinity, to variations of salinity or variations of temperature. Let's have a quick look to the catch of these species in the last 20 years. After a very rainy winter in 2019 and 2000, the species Venerupis corrugata, the catches of the Venerupis corrugata drop really dramatically, and the catches never recover. While the introduced species Ruditapes philippinarum, the Manila clam, the harvesting of these species start growing a lot during that years, and now it's one of the, it's the main species taking into account the amount of the tons that we are harvested every year. Okay, maybe we can think that it doesn't matter because what we were harvesting in the late 90s and what we are harvesting, the total we are harvesting now more or less is the same. But the problem is the price of the catches. The Manila clam, the introduced species, is less appreciated, and the price of this species is lower than the native animals. So for the fishers to get the same income, they must spend more time in the sea, so more fishing effort. So we will come back to this later. And what about for the other species, the cockle? Well, the catches of the cockle are very fluctuating, as you can see here, with the lowest catches after important rains, as the ones that happened in 2000, 2001, or 2006, or 2010. But on top of that, in 2010, 2011, the cockle got a parasite. It was infected by a parasite, Martellia cochilea, that almost extinguished the populations in the Rias Baixas, here where we are. So besides the ecological consequences, since these 4 bivalves are key species in the intertidal sediment flats, all this cause tremendous economic consequences. Changes in the price and value of the catches, higher fishing cost because fishers should work more time to get the same kilos, so more fishing effort, and is sometimes drop in the fishers' incomes. And although gender in fisheries are frequently not taken into account because fishing is usually seen as a male activity, and statistics frequently do not report the role of women in the harvest, in Galicia, intertidal shellfish harvesting is carried out almost exclusively by women. More than 5,000 are working in the intertidal. And because of that, because women are harvesting the species that are at the greatest risk with this extreme event or with these projections of the higher frequency of heat waves and heavy rains that are predicting the IPCC, for instance. So our hypothesis is that the impacts of the extreme events will be stronger in the intertidal bivalves than in the subtidal ones because of the flooding or the heating will be greater to the intertidal than to the subtidal. Of course, in shallower bivalves than the deeper ones, shallower and deeper here is the deep in the sediment, and in native bivalves than in the introduced ones. So if our hypotheses are true, there will be important implications for the small-scale fisheries with a bias against female fishers that are the ones that work in the intertidal bivalves. So the objectives of this project are to evaluate mortality and growth in different fishing beds, to evaluate the ecophysiological responses based on variations of temperature and salinity that are actually recorded in the field now, and also to work with the temperatures and the salinities that are predicting the models. To study the vulnerability of stress species to predators, to estimate economic cost and social changes associated with the predictions, including gender issues. And with all this information, we want to develop models for predicting sustainability of shellfish beds on short and long time scales, not only from a biological point of view, but also taking into account the socioeconomic point of view. But today I'm going just to talk about the two first objectives. So I already told you several times Galicia, so where we are. As Craig already told, we are in the corner of Spain. This is Spain, this is Portugal. You are just more than 8,000 kilometers far from me. And we are here, so facing directly the ocean. And it's a very special place, of course, not only because I was born here, but also because we have a very strong upwelling in front of our coast. So this makes that our areas that are fluvial valleys are similar to the fjords, but instead of being by ice, being created by ice, were created by rivers. But the structure is quite similar. So this upwelling that we have just in front of our areas makes all our area very, very productive, as it happened, for instance, in Chile. And also because it has this estuaric component in the inner part of the RIAs, we have places in the inner part of the RIAs where salinity or temperature varies a lot during winter or during summer. And in these places is where the most productive shellfish beds are. So we work in these 4 shellfish beds that are in 3 RIAs. The university, this is the Ria de Vigo, this is the National Park of Atlantic Islands, all this. The University of Vigo is here, and here is the marine station that I will show you in a picture a little bit later. So now in this second part of the talk, I'm going to present you some of the results that some of them has been already published, others are submitted, and we still have some of them in the oven we are still working on. And one of those is this one about mortality and growth in the fishing beds. So the results that I am going to present you are quite preliminary results, but I think that they are nicer to understand the other experiments. So to study mortality and growth in the fishing beds, we use these yellow baskets that we set in the sediment. We fill with sediment that previously was sieved, and we put 25 bivalves, the 4 species of bivalves, 25 of species, monospecific baskets. Here we have just cockles, for instance, and in the other one of species of clam, and so on. And all them were measured and marked with numbers. Then we put a lid on just to avoid predators. And every month we went to the field, we took the clams out, and we measure and count on for mortality. We did this during 2 years, and also we put eye buttons in the field to measure sediment temperature at 0, 5, 10, and 15 centimeters every 30 minutes. And also we use these small mini CTDs to measure water temperature and salinity also every 30 minutes. So this is the graph that I produced just for this talk. Is very preliminary graph with the mortality, the total mortality in each fishing bed. And as you can see, more or less, the mortality follows a similar pattern in the 4 fishing beds, well, at least in 3 fishing beds. This one, the orange one, follows a different pattern that we still don't know why. But just to, I'm going to comment just one fishing bed, Cambados, since all of the patterns are more or less the same. So we can see that one of the species, the native clams, Ruditapes decusatus, presented peaks, important peaks of mortality. One of those, the one with the red arrow, is mortality that we think that is produced by heat waves, almost one each summer. and an important peak of mortality in autumn that we think that was produced by heavy rains. While look at the Ruditapes philippinarum, remember that this is the species that is the introduced one, the Manila clam. So we hardly record any mortality, one or two animals. Regarding the other two species, Venerupis corrugata, that is very well appreciated here in well, not only in Spain, but also in Europe, we reach 20% of mortality associated with 2 heat waves and a flooding event, a heavy rain event. And Cerastoderma edule also present these peaks of mortality, but in this case, we are not sure that this mortality was produced by the extreme events or directly for the parasite Martellia cochilea that was with a very high how do you say this in English? Well, many of the cockles were infested by this parasite. So I think that we are not going to take into account this mortality because we cannot in the field. So you will see the results that we have in the lab, that the ones that we have in the lab were free of the parasite. So, because all these field experiments are always very complicated, since many variables cannot be controlled, as this one of the parasite, we ran several experiments in the lab with very interesting results. And in fact, some of the results are opposite to what we observe in the field. So now we are trying to connect both results, the lab and the field. Some of these results has been already published or already submitted. I put here the references just if you want to have a look to them. So these experiments were run at the marine station that is called ECIMAD, that the University of Vigo has in this island, Toralla Island, and this is the marine station where all of you are very welcome, of course. And to study the physiological responses of the adults to heat waves, we carry out the experiments in this mesocosm system that we had in the lab, where with these automatic pumps we simulated tides here automatically. Of course, we were not doing the tides. It was just automatically. So this is the low tide, excuse me, high tide, and this is low tide. So in each of these small tanks that were filled with sediment, we put 28 bivalves on it, and all tanks were replicated. Here you can see, for instance, very well the siphons of the Manila clam here with the low tide. So, and we did 4 different treatments with the temperature. One treatment was just the control, that it was trying to keep the normal temperature in summertime. Here, that is between 20 and 22. Then we have, excuse me. Then we have another treatment that it was 27, 32, and 33 degrees Celsius. We did these heat waves using these ceramic lamps that you have here during the diurnal low tide, when we just put the ceramic lamps close to the sediment. So here you can see the profiles, the temperature profiles of the 4 treatments. And the ceramic lamps were controlled by controllers, digital controllers, with feedback with thermocouples that were in the sediment, in the middle of the tank at 2 centimeters depth of the tank. The water temperature was also recorded with the mini CTDs, and the temperature in the sediment, besides the thermocouple, was recorded using also the eye buttons on the surface and at 5 centimeters. The heat wave happened during 4 consecutive days, only during low tide, and the measurements were done during before the stress, in the middle of the stress, day 2, at the end of the stress, day 4, and 2 days after finishing the stress that we call the recovery. What we measure? We measure 6 animals of each replicate and its treatment. And we measure, first of all, mortality, and then activity, burrowing activity, using pressure sensors that were set in these beakers filled with sediment and with 1 clam on each, that produce these burrowing, these spikes, that Sally Wooding, that is very, very patient, was able to know when the clams were burrowing or when they were just feeding or even pooing. It was impressive, the information that we got from these pressure sensors. Also, we measure oxygen consumption in these respirometers using the luminescent dissolved oxygen probes, feeding rate, clearance rate, feeding the animals with a no concentration of isocrasis galvana, measuring before and after 30 minutes of feeding. Look at that all the measurements were done individually. Also, we measure ammonium excretion and feces during 2.5 hours of each animal. So with all these parameters, we can calculate the scope for growth. Besides, when we finished with all these, measurements, we took samples for gonadal stage, and also with other animals coming directly from the experimental tanks. We took samples also for heat shock proteins. Because the experimental temperatures that I told you are temperatures at the surface of the sediment. But remember that these bivalves live buried in the sediment. So what we did was calculating the diffusion, the heat diffusion in the sediment to calculate the actual temperature that each animal was suffering, depending on the depth where they live. Also, the temperature was transformed to degree hours. So the increment above 22 degrees, that is the thermal optimum of this species per unit of time elapsed. And all these results are are presented in degree hours, although just for being more to be easier, I just put some special temperatures here. So what happened with mortality? With mortality, after 2 days of stress, that is the yellow one, the mortality was very high for Cerastoderma edule. That is the species that lives shallower in the sediment. In this sediment, we reach even temperatures of 41 degrees Celsius that we didn't want to arrive to this temperature, but sometimes the sediment really reached that temperature. And the mortality was really very, very high. Also, we had important mortalities of the species Venerupis corrugata at a lower temperature. Look at here. Here is 60 degree hours, but here we have important mortalities also at just 20 degree hours. And remember that this species is lives at 7 centimeters in the sediment. Then the next species in mortality was Ruditapes philippinarum, the Manila clam, that have not many. I mean, the probability of mortality was a little bit higher than 0.2 to 20% of mortality at approximately 38 degrees. And Ruditapes decusatus, the species that live lower in the sediment, deeper, excuse me, in the sediment, presented no mortality. In fact, although this 8 centimeters is the normal boreal depth of this species, we found it even at the bottom of the tanks escaping from heat, so at almost 13 centimeters. Regarding the burrowing activity, the behavior of these animals, we can see that Venerupis corrugata, this species, the activity diminished very a lot with temperature. In fact, the animals at highest temperature almost didn't move, while the contrary happened with Ruditapes decusatus. Here you can see that an increase of burrowing activity with the increase of temperature until they reach the temperature of 36, that the animals don't move. And this is because, as I told you before, Ruditapes decusatus escape because they have very, very long siphons and they can escape from heat deeper in the sediment. And this is an interesting response of Ruditapes philippinarum, they introduced the Manila clam, that seems that the the temperature is not influence the activity. So what happened with the scope for growth? With the scope for growth, we have two different responses. One that is when the temperature starts being higher, the scope for growth increase a little bit in these two species, Venerupis corrugata and Ruditapes philippinarum, or just stay stable as in the cockle or in Ruditapes decusatus. But as temperature increase, the scope for growth decrease in these three species. And at day 4, this is just already in the middle of the stress, just 2 days of stress. And the pattern is more or less the same at day 4, that is after 4 days of stress. And what happened with the reproduction is very impressive, because remember that we had the animals just 4 days with these heat waves. I mean, not just the heat wave. Remember that it was just in the low tide, not during the 24 hours. And we can see here that the cockle and Venerupis corrugata had an important reabsorption of the gonad in these 6 days compared to the day 0 that you have here. And even more, the Ruditapes decusatus, the deepest species, even when the gonad didn't reabsorb as in the other 2 species, the ova were broken, and some of the ova, or most of the ova, were atretic. So even at this these species that didn't show any effect during in the scope for growth, for instance, had this effect on its reproductive cycle. And Ruditapes philippinarum, the introduced species, didn't show any effect on their reproduction with the temperature. Okay, so in summary, we had important mortalities at the highest temperatures in the 4 species, except for the Ruditapes decusatus, the deepest species. But even when these species didn't show mortality, show changes or important changes in the burrowing activity and in the reproduction pattern. The other species already also have important reduction in the scope for growth and important problems with the reproduction, except for Ruditapes philippinarum that had mortality, lower scope for growth, but didn't vary the pattern of burrowing or reproduction. And what happened after the stress during these 2 days of recovery? We didn't record any more mortality, and the scope for growth was higher. We think that is a compensatory response that is produced by the previous stress. That's why the scope for growth increased a lot after the stress. But if the animal spawned, we didn't record an increase of scope for growth. So now let's move on to low salinity, to the results of the low salinity. So in general, in our latitude, the low salinity events happen during autumn, winter, and spring, the heavy rains. And but it's not the same if these heavy rains happen in autumn or in winter or in spring for the animals, because the reproductive cycle in general, except for Venerupis corrugata, in autumn, the clams are in the resting stage of the reproduction, and is starting gametogenesis in winter to reach spring in the already mature, ripe, and even spawning. So we wanted to know if a low salinity event would have the same effect in all these, in the three seasons, in these 3 seasons of the year. So that's why we ran this experiment 3 times, in autumn, winter, and spring. We use the same facilities, the same, more or less the same setting, but now with this pump, mixing the fresh water and seawater, we create 4 salinity ramps. One is 520 of salinity, another one 1025, 1530, and the control 3030. We choose the 30 salinity of 30 as a control, because this is the salinity in the fishing beds during this time of the year. We stress the animals during 6 days, and we let the animals 3 days for recovery. The system was the same. We simulate the tidal cycles of the natural tidal cycles, and the ramps were like this, like the ones that happen in the more or less in the ocean. So we measure the same variables as in the heat wave experiment, and at the beginning, the first time that we did this experiment, we did the measurements at the lowest salinities, so at 5, 10, 15, and the control 30. And what happened is that at those very low salinities, I mean in the beakers or in the respirometers, we had salinities of 5, 10, and 15, because the animals were coming directly from this stress. And what happened is that the animals just closed their valves, valves, and they didn't do anything. So they didn't feed, and they didn't breathe. So the scope for growth was 0, almost 0, but this was not the response that we wanted to get. So we decided to do to change the experiment and do the same, but after the stress. I mean, we stressed the animals, we took the animals when the stress finished, and we did the measurements at the highest salinities, 20, 25, and 30. And then the animals react and did things. So the burrowing behavior, we have 2 different patterns, Venerupis corrugata and Ruditapes philippinarum. At salinities below 20, they didn't do almost anything. remember that, for instance, Ruditapes, Venerupis corrugata, with the heat wave, they also they also closed their valves, and they didn't do anything. The reaction is not escape, but just shut down. And but when the salinity was higher than 20, the animals just move in the sediment. And Ruditapes decusatus, they stay quite inactive in the experiment, for instance, in March, and to recover the activity at salinities above 20 in May. Okay, so excuse me. So since these GAM diagrams, I mean, GAM is a very, very good and powerful statistical analysis for these models, but the graphs are quite complicated to look at. So I'm going just to choose some of them to explain you different results of that we obtained during all these experiments, and then I'm going to summarize everything, because I think that this is more useful than just have a look to all these diagrams. So, for instance, in autumn, Venerupis corrugata had low scope for growth at the lowest salinities, and even more, it didn't recover after the stress. So the scope for growth was very, very, very low in autumn. Remember that Venerupis corrugata was the clam that was in gametogenesis during autumn. The other species, we couldn't find any difference in the scope for growth at any salinity with the other species that are in the resting stage of development. In winter, for instance, Ruditapes decusatus had a very low, even negative scope for growth at the lowest salinity. And in spring, we have a very nice physiological compensation response. Look at this. At the lowest salinities, the animals had, for instance, this is Venerupis corrugata and this is Therastoderma edule. And both species have negative scope for growth at the lowest salinities. But at salinities salinities of 10 to 25 and 15 to 30, they have a high scope for growth, and look at the scope for growth of Therastoderma edule. This happened because they have a physiological compensation to stress. So the animal is stressed, not a lot, not a very, very high stress, because if they have very high stress, as in these ones, they just don't do anything. They have negative scope for growth. But when they are a little bit of stress, they are stressed, and they are in spring, so they are in the process of gametogenesis, in fact, late gametogenesis, and some of them spawning. Even when they are stressed, they have positive scope for growth because they need energy for the reproduction and for the stress, for fighting against the stress. And it's a very nice response. So in summary, what happened? Okay, so remember, when we measure under the ebb-tide conditions, so at the lowest salinities, the animals just close their valves, so the scope for growth was near 0, so they don't grow, and they also don't burrow, and the consequence of not burrowing is that potentially they can be predated, because they are going to stay on the surface of the sediment. And at the lowest salinity ramps, we have similar consequences: negative scope for growth and lower burrowing. So again, no burrow and no predation. And at intermediate salinities, all species have a higher scope for growth in spring when they are in reproduction. So they show this physiological compensation. But they have lower burrowing, so maybe they can be predated. And at the highest salinity ramp, not the control one, but the 1530, the only species that was affected was Venerupis corrugata, again with higher scope for growth and a lower burrowing activity. And now I am going to move fast through the physiological responses to low salinities of juveniles. This experiment was done just with one species, Terastoderma edule, and we didn't use sediment because it was very difficult to separate the settles from the sediment to do the measurements. So we decided to do the experiment just in the water. So we got two different sizes: the thread drifters, that are those that are that have the primary settlement, that are very small, 3.7 millimeters, and the sedentary settles, that is the secondary settlement, that they are already 9.6 millimeters. We stress these animals with 8 salinities and with 2 levels of stress, 2 days, looking for an acute response to the stress, and 7 days that we were looking for a possible acclimatization to the stress. And also we look for a long-term, a possible long-term effect. So we measure the same: mortality, oxygen consumption, feeding rate, ammonia excretion, and growth. And what we found was a different response depending on the size of the animals. The drifters, so the small ones, start feeding at salinities of 10 and even more at 15, while the sedentary settles, the sedentary settles don't feed until they are at salinities of 20. This pattern is more or less the same in the oxygen consumption, and also in the oxygen consumption, excuse me. And regarding the growth, these sedentary, the small ones, the drifters, grow above salinities of 20, while the sedentary settles, we couldn't find any significant difference in growth. So after 7 days of stress, what happened is that all animals kept at salinities of lower than 15 died, in fact died at day 4. And the ones that survived, they show different response depending on the size. The thread drifters were acclimated to low salinities. In fact, we couldn't find any difference, for instance, in the clearance rate or in the excretion rate or in the oxygen consumption among salinities. While the sedentary settles, the big ones, are not acclimated to the low salinity. They have very low clearance rate, or of course, or of course a high excretion rate or high oxygen consumption due to the stress. So what we conclude is that the thread drifters are more aerialine than the juveniles, because the thread drifters occupy less stable environments and requires more flexibility to the changing environmental conditions and to optimize the energy as fast as possible to be able to grow to the refugee size in the sediment. And we wanted to go further and see if the effects of the stress stay longer, for long periods. So we took the thread drifters, we stressed them during 2 days at 4 different salinities, and then we put them in the field, in these baskets hanging in the marina of the marine station. And we measure growth at the end of the stress, after 5 days, 1 month, and 10 months, and gonadal development at month 10. And what we saw is, of course, that the animals that were at salinity of 5 died. And after 2 days of stress, that is the gray bars here. After 2 days of stress, the animals at the low salinity grow less than the other ones. After 5 days, the animals at 35 grew more than the other ones, but after 1 month, that are the green bars, after 1 month, the animals recover, and we couldn't see any difference among treatments in the growth. But surprisingly, at least for us, after 10 months that these animals reach the maturation in this time, after 10 months, we saw difference in the gametogenesis. Look at these graphs here. The animals that were stressed at salinities of 15, they follow all the gametogenesis processes but they were slower than the animals that were stressed at salinities of 20, 25 and 36 and 35 that were already spawning. So something that happened, and 2 days of stress that happened 10 months before, we can see the effects in the reproduction, and the effects were really very clear, and not, for instance, in the size. So now I'm going to just to finish asking you the question that I told you at the beginning. And now what? What do we do with all these results? So I just summarizing, high temperatures provoke significant mortality in 2 species, the Cerastoderma edulis, the cockle, and Venerupis corrugata. Decrease growth potential for all species, and particularly for these 2 species, and diminish the ability to burrow, so more exposition to predators. And salinities equal or lower than 15 don't produce mortalities, big mortalities. But the problem is that decrease a lot the scope for growth. So what happened in the field? For instance, this is one of the fishing beds. So in autumn, 90.5% of the time, that fishing bed is under salinities under 15. In winter, 21 of the time, and in spring, 23 of the time. So during this time, the animals just close their valves and they don't do anything. So, well, doing some calculations that we already published, those animals are losing more than 20% of their scope for growth. So their ability or the potential to growth. And this is a lot. I mean, it's 20% less of the growth, and even more in some some months of the year, because the reproductive processes, some animals are even in negative scope for growth, even without stress. So with stress, this scope for growth is even more negative. So all this is an increases the probability of energy deficit of all these animals. So this generates less growth, so more time to reach the commercial sizes, for instance, less energy for reproduction, not only for growth, but also for reproduction, and less energy to defend against, for instance, parasites, or to other stress like pollution, for instance. So even when the mortality is not the main cause of these salinity drops, we can have important consequences for not only for of course, for the ecosystems, because remember that these species are key species in these tidal floods, but also for the fisheries. So thank you for your attention. And I think that now of course, I would like to have your inputs and your comments and trying to answer this question. What do you think that can happen with all those results? Of course, for the ecosystem and also for the fisheries. So now I'm going just to leave the screen. Okay.
Okay, thank you very much, Elsa. I'll ask everybody to turn on their videos again, and then we will we'll give you a round of applause for a beautiful talk.
Thank you.
So we're running a little bit late. Some of you may have to leave the meeting at this point, but we'd be very happy to have, I think Elsa would be very happy to have input from you and any questions that you might have. Does anyone have a question for Elsa?
Yeah, and if you don't have time now, you can just send me an email. You have my email in the first slide of the talk, and just send me an email asking what you want. I know that you need to do a summary or something like that. So
that's right. Some of the students have to do a summary. I have a question for you, Elsa. I'm interested in knowing how you actually documented the resorption and the spawning in these experiments, and how you used histology to do that. Was resorption just because you saw a breakdown of oocytes in the gonad?
No, because we saw the reabsorption. So we did the histology, the classical histology with eosin hematocylin. And if I share the screen again, in fact, in one of the slides, here, correct. This is the reabsorption of the resorption of the gonad. It's after spawning. This is a natural process. When the animal after spawning and at the end of the reproductive season, the gonad started being reabsorbed by the hemocytes, and also with the atresia, well, we can see the atresia ova. And the question is that these processes are, we could see the reabsorption in more animals at the highest temperatures. So in fact, at 37, almost all the gonad was completely disorganized.
Yeah, okay. I thought it was quite remarkable that this happened so quickly after just
a
few days. Very interesting.
It's very interesting. And also we have I'm working now on the publication. In fact, I think that is going to be the next one be sent. And not only because here I just present the ones with the heat wave experiment, but also we saw this with the low salinity events. We saw difference in gonad, and it's just in 6 days. In 6 days in the case of the low salinity, and in 4 days in the case of temperature. And it's not that we were cooking the animals all the time. I mean, we had this a temperature ramp, and the rest of the time the animals were just at normal temperature. And yeah.
Okay, thank you very much. Does anybody else have a question for Elsa? Nancy Treneman.
Hello, and thank you very much. That was really, really interesting. I work with shipworms, so they don't burrow in mud, but they burrow in wood, and I'd be very interested to see how they respond to the things that you've been doing. I had some questions. In your images of the harvest, it looked like a very intensive harvest effort. Is that
Yes.
all day, all year? Wow.
No. Okay. They organize, each fisher guild organize together with the administration the days that they can harvest, and the limit is the kilos that they can harvest every day. They go during low tides, not all days. They go during low tides, and they harvest 5 kilos of each species, or depending on the density of the animals, they harvest 2 kilos of one species, 5 of the other, and 10 of the other one.
Right.
So
it's a limited harvest.
Excuse me?
It's a harvest that's controlled. It's limited.
Yes. They have a control point when they go out of the fishing bed. They pass through a control point where they wait and control. They have automatic machines that control the sizes and the weight of each species. Yeah, it's an artisanal fishery, because you see how they do the harvest with very, very simple instruments, devices, but then they have a very sophisticated processes for control the fishery. That's why we think that it's a sustainable fishery, because it's very, very well controlled here in Galicia.
Is there, in looking at your data, you're looking at climate change and seeing that these events are going to be more extreme and maybe have lower reproduction and higher predation, not by humans, by other predators. Is there some ideas about either having beds that are not harvested as kind of a reserve, or changing the season at which you harvest so there's a greater chance for reproduction? Are those some things you guys have thought about?
And in fact, this effect on reproduction, some of the women that when we go to the field, we are with them, and in fact, during the reproduction season, they don't harvest the species.
Okay.
Well, more or less, but yes, they try not to harvest that species, because even they have the species have low market size, because after the animals are collected, they go to at least during 2 days to how they call this in English, the place that you put the bivalves to circulate water and to get rid of bacteria.
Yeah, just put them in an aquarium and it clears out.
Well, yeah, big, huge aquariums. Before going.
Yeah, depuration, I think that's called.
Depuration, okay. Exactly. Before going to the market. And if the animals are really very ripe, they spawn in the depuration, and when the animals arrive to the market, they are almost empty. I mean, they are not as big because they already spawn in the aquarium. So they try not to collect the animals when they are very, very ripe.
Okay.
But yes, the consequences of all these changes are going to be quite important in this small village, because many people live from harvesting these animals, and we are seeing already a shift in the species between the native species and the Manila clam, the introduced species. And is not, I mean, they sell these species, but the price is twice cheaper as the other ones, because it's not so appreciated as the other ones. Yeah.
Thank you. It's very interesting. Appreciate your answer.
Thank you.
Yeah.
Okay, we've been going for quite a long time. I think we'll end your lecture now. We'll give you one more round of applause.
Yes.
And we really appreciate it. It's nice to see you.
Thank you. Thank you for listening.
Now you can go have your dinner, and I hope you eat pulpo for me, a nice octopus, which is food dishes in Galicia.
Pulpo is one of the problems now here in Galicia. Yeah. There is no more pulpo, almost. The octopus.
I'm sorry to hear that.
Loves octopus. So.
Okay, thank you everybody for attending, and thank you also for a very nice seminar.
Thank you. Thank you.
Thank you.
Bye.
Bye-bye. Huh, how nice.